Good day, everyone. You are welcome to today's practical class. Today, we shall be looking at plant anatomy. Another plant anatomy, that aspect we shall look at is internal structure of dicot and monocot stems and roots. So we're going to look at how to prepare the transfer sections of the stems and roots. So I have stated there so that you'll be able to follow the instruction very well. Our major aim is to prepare a staged month of transfer sections of dicots and monocot stems and roots, so as to enable us to be able to study various tissues. What are those things we are going to need? That is the apparatus. We are going to need a young dicot plant, something like a water leaf, that is a young one that we can easily cut. Also, a young monocot plant, for example, maize or grass, we need microscope, sharp blade, preferably a new blade, glass slides, petri dishes, cover slips, hyoscine. Hyoscine is a stain agent. We need spatula, we need blotting papers. So how do we go about it? In the procedure, we are starting with dicot stem. After dicot stem, then you repeat the same process for dicot root as well as monocot stem and monocot root. Now the first thing you're going to do, we're going to remove the leaves from the stem and bring out the stem. We cut out the stem reasonable length that we need. Hold the stem between the index finger and the thumb. This is to enable us to cut out the section. So now take the blade in the longitudinal side of the plant or the stem. Then you cut several slices of thin sections. You have to be careful when you are cutting this with the blade. While you are cutting it, you fill a petri dish with water. You use the edge of the blade to drop the sections into the petri dish with water with the help of a spatula. That is, when it is not dropping, you use the spatula to move it down from the side of the blade into the petri dish with water. Then you fill a different petri dish about half full with hyoscine, your staining agent. Then pick 2 or 3 thin or good, that is, when we say good, perfectly round transfer sections, that is those ones that are not cut in the process of cutting it, that are not like semicircle, those that have full circles. Now you place this in the petri dish with hyoscine. Let the sections rest in the stain for a couple of minutes. That is, you allow the sections to stay in hyoscine for some minutes. This is to allow the hyoscine to stain the sections. That is, for the sections to be stained. So after a while, you drain the sections of the stain. That is, you remove the stain and you rinse with water. You are rinsing with water so as to remove excess stains. It is believed that the tissues must have been stained so as to enable us to see the different tissues that are present in a plant. Now, in the stems of this plant, then with the help of spatula, remove a stained section. Place it in the middle of a glass slide, then put a little water, and with the help of a needle, carefully use the cover slip slowly to cover the section on top. It is possible to have excess water around the cover slip or at the edge of the cover slip. Place a blotting paper. That blotting paper will soak the excess water. The essence is to ensure that when you are looking at it or observing this section under the microscope, it is not going to be water that you will be looking at. So ensure, while you are covering with cover slip, ensure that you didn't cover with air bubbles. That is, air bubbles are not formed while you are mounting. Then place carefully on the stage, that is on the stage of a microscope. You place it carefully there on the stage of a microscope and observe. You draw your observation, that is, whatever you see, you draw it. Then you repeat this procedure, repeat the procedure with dicot root, thereafter with monocot stem and monocot root. In the same way, you draw your observations. And in that case, that means you are expected to have 4 different diagrams. Thank you so much for listening.